Hi, today we're going to talk about the first section of Chapter 1, which is what is science? So think about it. One day long ago, someone looked around and wondered, where did plants and animals come from? How did I come to be? Since then, humans have tried to answer those questions in different ways. Some ways of explaining the world have stayed the same over time. However, science is always changing. So, our essential questions today are: what role does science play in the study of life? And our objectives are to identify the goals of science and to list the steps that scientists take to solve problems. Here's some of the vocabulary to be on the lookout for throughout this lesson. Please be sure to check out the virtual flashcards and the other resources that we have in our course page for Chapter 1. And be sure to take notes while we complete this first lesson. What is science? Well, it's an organized way of using evidence to learn about the natural world. We're trying to understand, explain, and then use those explanations to make useful predictions. Science isn't just a collection of facts. Scientific ideas are open to being tested, they're open to discussion, and they're often open to revision. So science is constantly changing, and that's a good thing. It's because we're always learning more and we're always exploring new possibilities. When we look at the scientific process, the first thing we always need to do is to make observations. And we do that by using our senses. It's what we see, what we taste, what we hear, what we smell. So an observation is the process of gathering information about events and processes in a careful, orderly way. So for instance, someone might notice, well, I'm looking at grasses in two different marsh areas, and I noticed that the grasses in location B seem to grow much taller than the grasses in location A. Well, I may infer, using my prior knowledge, is it possible that the grasses in location B have some type of nutrient in the soil that's lacking in location A? If I had enough prior knowledge, I might even realize that this could possibly be nitrogen. Now I'm using an inference. I'm using a logical interpretation based on prior knowledge and experiences. I may then even make a hypothesis. Marsh grass growth is limited by available nitrogen, meaning if there's plenty of nitrogen, the grasses grow very tall, and if there is not plenty of nitrogen, they do not grow very tall, so it limits their growth. Hypothesis is a little bit more than an educated guess, like you may have learned in previous science classes. It's a proposed scientific explanation for a set of observations. You're using your prior knowledge, your inferences, your imagination. It's also really important that a hypothesis be testable. We have to be able to test it so that we can collect data. As we collect data, we may go back and we may support a hypothesis, we may rule out a hypothesis, and we may even have to revise a hypothesis. When we design controlled experiments, we look at different factors, and we call these factors variables. So for instance, the temperature, the light, the time, the availability of nutrients, these are all variables that are affecting the growth of the marsh grasses. For a hypothesis to be tested, it's important that only one variable be changed, and we call all the other variables controls. This type of experiment is called a controlled experiment. So let's look at some of the different variables. It's important to control variables because if several variables are changed in the experiment, it's hard for us to tell which variable is responsible. For instance, which variable is responsible for the growth of the grasses if there's all different factors playing in. So we always try to focus on just one variable. That one variable that's deliberately changed, we call the independent variable. Sometimes it's also called the manipulated variable. The variable that is observed and that changes in response to the independent variable, we call the dependent variable. Sometimes that's also called the responding variable. Usually, in an experiment, we divide whatever it is we're testing, the marsh grasses for instance, into two groups, the control group and the experimental group. The control group is exposed to all of the variables with the exception of that one independent variable. The experimental group is exposed to all of the exact same variables as the control, as well as the independent variable. Let's take a look at our experiment. We've got our two sets of marsh grasses. They're going to have the same soil type, the same plant density, the same amount of water. The difference here, the independent variable, is that the experimental group is going to receive nitrogen added to the soil. The control group will not. We then are going to collect and analyze our data. Well, the control group, we can see here, has significantly less growth than the experimental group. We collect data in two ways, quantitative data and qualitative data. Let's look for a second at the difference. Qualitative data is descriptive, and we describe things using characteristics. But sometimes qualitative data can be a bit vague. It's like saying the grasses in the experimental group grew very tall. Well, what's tall to one person might not be very tall to another. Quantitative data, on the other hand, we're using numbers. We're counting, we're measuring. Now, quantitative data may be that the experimental group grass grew 12 centimeters. We're giving it a quantity, a number. That's the difference between those two types of data. We usually take our data and we analyze it in some way, and usually that's in the form of a graph. And we use this to get a good look at our data so we can really draw a nice conclusion. So here we have the height of grasses, the height in meters over a time in weeks. We can see that the control growth is in blue and the experimental group's growth is in red. We can see here that the experimental grasses grew much taller over time. This is allowing us to draw conclusions that it looks to me like our hypothesis was certainly correct. Adding nitrogen to the soil certainly increased the growth of the grasses. There are times sometimes, though, that our hypotheses will not be correct. And if that's the case, often we have to go back, create a new hypothesis, a new experiment, make new observations. So keep in mind that hypotheses may need to be revised. New experiments might need to be completed before hypotheses can be supported and new conclusions can be drawn. So thanks for listening today to Section 1 of Chapter 1. Join us again for our next lesson on Section 2 of Chapter 1. If you have any questions, please see myself, Mrs. Graham, or Mrs. Kroll for help, and we hope to see you in our next live class.